Luria Delbruck experiment aimed was to know whether the mutation in bacterial culture is spontaneous or adaptive. In nature, mutations are rare events which occur at random and arise spontaneously with no regard to environmental conditions. Spontaneous bacterial mutations may occur at a rate of only one mutation in 1 million bacterial cells to a rate of only one mutation in 10 billion bacterial cells. Generally, the mutants in a cell population are masked by the greater numbers of unmutated cells. Isolating a mutant cell is like looking for the proverbial needle in a haystack. However, microbiologists have developed techniques which facilitate isolation of the few mutants from a large population of non-mutated wild-type cells. For example, an antibiotic can be incorporated in a medium to select for antibiotic-resistant mutants. For a long time, many geneticists were very reluctant to believe that the observable changes in bacterial cultures were ever anything more than physiological adaptations or phenotypic changes. That is, they refused to believe that bacteria have stable hereditary systems and could undergo permanent changes or mutations, and assumed that any changes in the characteristics of a culture were simply due to environmental influences. However, Max Delbruck and Salvador Luria believed that bacteria have stable hereditary mechanisms, and in 1943 they performed an experiment that proved the point. Bacterial viruses called bacteriophages are capable of killing bacteria. When susceptible bacteria are exposed to a phage, some of the bacterial cells survive, and they and their descendants are resistant to the phage. Let us suppose that resistant cells are the result of contact with phages. Then if we set up a large number of identical bacterial cultures and expose them all to identical batches of phage, approximately the same number of resistant cells should appear in all cultures. However, if the resistant cells are really the result of mutations, and since mutations occur entirely at random, then when we grow many identical cultures and expose them all to identical batches of phage, we find a great fluctuation in the numbers of resistant cells. In some cultures, there may be no mutations and therefore no resistant cells, but in other cultures a mutation might have occurred very early, so that nearly all the cells are resistant. Some microbiologists assumed that these cells were modified by their contact with the phage, so they became resistant by physiological adaptation. But Luria and Delbruck believed that phage-resistant bacteria were the result of mutations that occurred before the bacteria came into contact with the phages. Hypothesis 1. Mutation hypothesis. There is a finite and small probability, A, that a mutation from susceptible to resistant will occur at a constant rate. Hypothesis. Adaptation hypothesis. There is a finite and small probability that each cell in the culture might survive the phage attack, and all subsequent progeny from survivors will be resistant. Hypothesis 1 will generate resistant clones, and their prevalence will be determined by how early in culture growth the mutation event occurred. The proportion of resistant bacteria will increase over time, prior to selection. If you study a large number of small cultures, the proportion of resistant bacteria in each culture will vary or fluctuate wildly, depending on how early the mutation occurred. Hypothesis 2. No clones. Some bacteria will survive and reproduce resistant daughter cells. Selection begins only after the addition of phage, not before. The proportion of potential survivors resistant bacteria will remain constant. If you study a large number of small cultures, the proportion of resistant bacteria will remain constant, size of the culture is irrelevant. Formation of phage-resistant bacteria by mutation. In some culture a mutation occurs very early, and thus many of the cells are resistant. In others mutation may occur very late, so that there are few resistant cells. In some there is no mutation and hence no mutation. Luria and Delbruck found a much greater fluctuation in numbers than could be accounted for physiological adaptation, and thus they proved statistically by their fluctuation test that phage resistance was really the result of mutation.